In this online lecture, we'd like to talk about three-point mapping. Basically, we want to go through some of the setup for how we do three-point mapping, and then we want to work through one example of a three-point mapping problem. Now, so far you've worked with two-point mapping, as we call it, meaning that we use two loci, and this was primarily used for chromosome location, where we would have a known site on a chromosome and then ask, okay, we have a new mutant. Does it belong on chromosome 1, 2, 3, or 4 of a fruit fly, or what other organism you might be working with? Two-point mapping provides us a way to look both at location and relative distance between the two loci, although we are generally limited to 50 map units being the maximum that we can identify between two loci. With three-point mapping, we add some other variables. We can again talk about distance, but this time the distances can be greater than 50 map units. We also can introduce the idea of order of the loci. Do they belong on top, bottom, middle? Where does the new, say, mutant locus belong? I've shown here locus 1, 2, and 3, trying to identify where these loci are found. Now, why do we do this to you? Why do we bring this forward in most classical genetics courses? Well, it is certainly a classical genetic technique. It was the way that new mutants were, once they were identified, were located on fruit fly chromosomes. It was also used for many other species. So it's a general technique that's important to know. It's also a capstone exercise, we'll call it. As you know, we worked you up through the preceding labs so that you started out with basic modes of inheritance, very simple crosses. We then introduced more complicated modes of inheritance. And then finally, we've introduced mapping in the way of two-point mapping. To do a three-point mapping problem requires your knowledge of all of these elements, plus you have to be able to think about Mendel's first principle, the principle of segregation, as well as Mendel's second principle, independent assortment, because clearly you're violating the idea of independent assortment. You also have to be able to do trihybrid crosses. So to be able to do a three-point map successfully requires a large number of skills that be operating at the same time. So it makes a very good experience and a way to sort of see how well we're doing at this point. Now, how do we do a three-point cross? Well, first we need to talk about the 3 points. We're talking about a trihybrid cross. As with other mapping, we will always use one standard cross here. This will be a back cross. In this case, we're going to look at 3 different loci, so we'll call it a trihybrid back cross. By back cross or test cross, we mean that one of the parents will always be a complete recessive or a completely opposite form of the alleles, in case that you're using a dominant as one of your markers in your trihybrid. But for this, we're going to we're going to use one individual who is a complete trihybrid heterozygous at all 3 loci, and we'll be recrossing it with a complete homozygote for the alternate or rare alleles. Now, when we do this, of course, we have a very simple outcome. It's one of the reasons we make this particular cross, is that each of these segregation ratios is a 1-to-1. We don't have to worry about a 3-to-1 or a 1-to-2-to-1 or any other complicated ratios. We simply can do it as a set of 3 1-to-1 base ratios. Now, when we put all these together to give our final genotypes, we're going to expect to have 8 different genotype classes. And sure enough, we do. These are the examples that we have. You would expect to see some of our genotype individuals be big A, big B, little b, big C, little c. You'd expect an equal number to be little a, little b, little c, little c. There will be 8 of these classes. That's all you will ever have from this cross, and that's the only type of cross that we will use doing three-point mapping. Now, as you know, in lab and other places, we don't generally use big A, big B, little a. We tend to use wild type as plus, and then designate the other alleles by their appropriate form. Whether if they're a dominant, you would use a capital letter, a big A. If they're recessive, you use little a. Here we're pretending that all 3 mutants that we're interested in, A, B, and C, are the recessive forms. So I'm going to use this standard sort of symbolism that we have here with the pluses, and that'll continue with that. Most problems in your textbook and elsewhere also use this, so that's the best reason to employ it. Now, it doesn't change anything as far as the outcome. You're still going to get a 1-to-1 segregation ratio for each locus, and you're going to get 8 parts, all 1-to-1 to 1-to-1 to 1-to-1. The only difference will be the big A's will be replaced by a wild type or plus. The big B's will be replaced by that as well. So something you just have to keep in mind. Now let's remember that we're dealing with linkage. So in this case, we know that all 3 loci are on the same chromosome, on the same homolog. So we want to designate them in this form. We'll put, in this case, wild on one homolog and A, B, C on the other. These little lines in the middle representing some stylized chromosomes. Now we want to concentrate on the heterozygous individual. And just like we did with a two-point cross, this is where all of the action is going to take place that we can detect. We're looking for crossover and then recombination between these two different homologs. We certainly have that going on in the test cross individual, the little a, little b, little c, trihybrid recessive, but you're not going to see any recombination there. You'll see crossover, but you won't see any differences. All the action that we're going to see is in this one individual that is our trihybrid. So we're going to concentrate on it. In fact, later on, we'll completely remove pretty much the input that we get from the recessive individuals. And lots of problems actually work simply by asking you, here are the gamete types being made by this trihybrid heterozygous individual. Let's use that to do mapping, which you certainly can do. Now, here's just an example of what we're talking about. Here are those progeny types that we had earlier. The little arrows that you see, the three arrows, indicate the contributions by the recessive individual, the test cross individual. If we simply remove those, they have no effect. They're merely there so that phenotypically we can see what the outcome is. You get two little A's together, you'll see the recessive phenotype. You get two little B's, you'll see the recessive for that, etc. So you need those to, of course, make it where you can see the phenotype. But as far as mechanically working the problem, this side of the equation is not that important. Now, we do need to point out that in the way I've drawn it here with these two little lines for this individual, we are only talking about two of the chromatids. Remember that we always have four chromatids available during meiosis, during synapsis. All of this crossover and recombination occurs during the early part of meiosis once you have synapsis occurring of your two homologs and your tetrad, as we call it, your four chromatids. So we're actually representing this as a tetrad. We know it to be present, but for simplicity, we're just going to concentrate on those homologs that might in fact, or chromatids that might in fact crossover. Be wary that there are two other forms here. There's another plus chromatid. There's another little a, little b, little c chromatid. This matters in certain problems when you have to go backwards and you have to count. And remember that each time you have one of these events, you also have to count in these other chromatids. But for us, we're going to simply concentrate while we're learning to do three-point mapping on these two forms, and we'll just pretend that the others are there, but we're not going to look at them. Okay, now let me start with a little problem. The problem will be where we will assume that the correct order at this point of the loci is ABC. We'll make it simple. We'll also assume that all three recessive alleles are on one homolog. Little a, little b, little c are the recessive forms, and we've put them on one homolog. We'll see how that differs in just a moment, but for right now, let's work with that. Now we're going to have two types of single crossovers, plus something new for us, which is a double crossover. So let's look at how we would get those. Now, first thing we're going to do is look at no crossover, because that's obviously the most important one here. So if we have no crossover at all, simply go through meiosis, there's no exchange of material between paternal and maternal homologs, then we would expect to see the gametes from this are very simply those that carry plus, wild, and recessive, little a, little b, little c. Those would be the gametes. We call these no crossover the parental or parental-like. It's just a common name. And very importantly for you, we expect these to be the majority of the progeny for two reasons. One, the most common thing to happen is no crossover. Also, you've got those other homologs out there which are not crossing over at all. So most of the progeny are going to look like the genetic configuration of the parent. Now, what if we have a crossover? Well, let's make a crossover now between the area of the A locus and the B locus. So on the left side of this particular chromosome. What I do when I'm learning to do this was I sort of take my pencil and maybe draw a line that represents where the crossover would be. In this case, we would start on the top with a wild or plus allele. We would cross over to the other homolog, the other chromatid in this case, and we would grab the little b and the little c component. That would be where the crossover would occur. That means that we're going to have one gamete that has a plus or wild type, then goes and picks up a B and a C. At the same time, since crossovers work in both directions, the other chromatid direction would be little a being picked up from the bottom chromatid, crossing over and now grabbing the wild type and another wild type at the B and C locus. So that our single crossover gametes, giving the starting point, would be A plus or A wild or plus BC. That would be one pair of crossovers. If we cross over on the other end, you can do the same thing, take your pencil and follow along, but this time you would have plus plus cross over to C, or AB cross over and pick up wild or plus. That would be your other set. That's a single crossover. Okay, but now what's going to happen is that you could have both of those crossovers occurring at the same time. It's not going to be very common. It'll in fact be the least common outcome, but you will see it, and so we need to recognize what it would look like. So here's what we would have. This would be a double crossover. You would cross over from wild on the top, cross over and pick up the B locus at the bottom, cross back to the top, and end up with a wild type allele for your third locus. The corresponding homolog would now have a little a, cross over, pick up a wild or plus, cross back and pick up C. So what you've done now is literally move the alleles that were found in the middle locus. You have moved them from one homolog to the other by recombination. Again, these are going to be your least likely out. Now let's look at all of these together, and we see that indeed we have 8 outcomes, but we've grouped them in groups of 2, so we have 4 pairs. The first one in this case being a no crossover. That would be the most common set of progeny that you have. You have a double crossover, which is at the bottom, which will have the least number of progeny, and then you would have 2 groups of single crossovers, and the number of progeny related to these single crossovers, just like it did in a 2-point map, depends on how far apart A is from B and how far C is from B. Those will determine the map units or the number of times you get recombinants here. Now, what would happen if we change the configuration? Notice we have not changed the genotype of the individual. It's still A plus B plus C, or big A, big B, little b, big C, little c, if you're using those letters. We haven't changed that. We've simply changed the starting configuration of that trihybrid heterozygous parent. This time on one homolog, we have A, wild, and we're starting with wild BC on the other homolog. Now this is actually a more realistic configuration, because very often what we have is 2 known sites, 2 known loci that we can work. We'll develop a new mutant and we'll wonder, okay, where does we mapped that mutant to the same chromosome, but where does it actually fit in relation to these existing loci? So this is a very common and realistic configuration in most systems. Now what's going to happen? Well, you're still going to get 8 outcomes. You're going to get 4 pairs. You're going to get the same 4 pairs, except now, because you're starting in a different configuration, a different set of pairs will be the most common, the least common, and the relative crossovers. This is a fairly difficult concept to work with, so it's worth your time to kind of play with it a little bit. Draw out a few of these configurations yourself and see what you would get. But in our case, the most common thing is no crossover. Parental, we're going to get A++, which is the top one, plus BC, which is the bottom. We're going to cross over every now and then between A and B, which will give us +++, and the resulting A, B, C, which is the first single crossover. We'll get another single crossover on the other end, and then finally we get this sort of odd arrangement for us, but remember, we're moving B in a double crossover, so it's not odd in that sense. It's just different from what we saw. We now have A, B plus and plus C, because we've moved the B from being associated with the C on the bottom to with the A on the top. And to do that required 2 crossovers, a double crossover. Now you can see again that the ends didn't change. We still moved on the one single crossover the end toward the A. We moved on the other single crossover the end toward the C. And finally we moved the B. Okay, now some rules for how we actually do 3-point mapping. I thought I would break this into a few simple steps, and that way you could always follow these steps to move you through the process. Okay, you can make up your own, certainly. You can add to these or whatever you need, but these are a starting point. First thing you want to do is you want to pair the offspring phenotypes. When you receive your data, you're just writing it down. You're really not thinking about how they group together, but you will need to pair them. We're going to show you how to do that. You then need from those pairs to determine the parental-like or null crossover. By definition, this will be the pair with the largest number of progeny. You then determine the double crossovers, which by definition will be the smallest number. Now I want to say something here. You need to be very careful, because in problems in textbooks and problems in other places, you may not get any double crossover progeny. So you may only have 6 classes of progeny. If that happens, that means you're missing 2 individuals, and they're simply 0. You didn't receive any. And if you don't receive any, you don't write them down. So some problems will have all 8 and it'll indicate 2 of them as 0, the smallest. Other times they'll simply leave those out. And if you see 6 offspring outcomes, you got to know there's supposed to be 8. So you'll add those other 2. There are lots of problems like that, and you should try working some. Once you have those 2 determined, you can then determine the order of the loci by comparing parental with the double. You're always going to compare everything to this parental. So once you define that, pair is a very important pair. If they're out of order, and they may be, you may have written them down A, B, C, but in fact the real order is A, C, B, or something else. You've just written the phenotypes down, tall, curly, smooth, or something, and really it's supposed to be tall, smooth, curly. Well, that's okay. We're just going to reorder them. It's important to reorder them, though, because if you don't, it makes it a mental gymnastics to try to do the rest of the work. Then you simply calculate up some mathematical values, the actual values for single crossover and double crossover. It's done by simply taking the number of offspring divided by the total for each of these classes. We'll do that quickly. And then finally, to get our map units, once you've got the order between the single crossovers and the ends or other forms, you're going to add the single crossover to the double crossover, because remember, map distance is how many times did I get a crossover between two loci. Well, if there was a single crossover, you would add those, of course, but the doubles also involved a crossover between those two loci. So for more accuracy, which is one of the things we get from a 3-point cross, you want to include this. All right, let's try a problem. I've set up a classic F1 trihybrid test cross where we've got black short wavy times a white long straight. Could be anything. You're told about the modes of inheritance. Again, in lab, these will be much more difficult modes of inheritance. It's a bit more of a challenge because you have sex linkage, you have lethals, you have dominance, you have codominance. But here we're going to just try to show you the basics using very simple, straightforward modes of inheritance. So black's dominant over white, short's dominant over long, wavy over straight. Okay, we want to symbolize these individuals. So we set them down as plus A plus B plus C times little a, little b, little c. So classic little form there. The offspring that are observed, we're given this as data, is that we have white short wavy, 46 of those, white short straight, 20 of those. You can read down through here. Okay, now what's the first thing we want to do? The first thing we had on our list was to pair the offspring genotypes. How do we do that? We're simply going to do that by their numbers. Things with similar numbers should pair up. So in our case, white short wavy at 46 pairs with black long straight, 42. Those are the two closest numbers. Others that pair would be white long straight and black short wavy, which are both have comparable numbers. And then you have to kind of work between the others. You got a white short straight at 20, a black long wavy at 20. Those should probably fit together. We'll show you a technique to double check this in just a moment, but you just basically pair them up. You then want to write them down in these pairs. I tend to write the largest pair first, then I write the smallest pair underneath that, and then I pick either of the other two to write them in some order below that. Be sure to include their genotype arrangements, A plus 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 BC beside each one. Now, one of the things you'll want to do is you'll want to double check that you do not have two of the same phenotypes or two of the same alleles in one of your pairs. You can't, in this case, have a black long wavy and a white long straight. If you do, something was wrong. Now, that's not the way the data looked here, but if you had come up with that, two longs, you've got to have one of each thing. You've got one black, one white, one short, one long, one wavy, one straight. Okay, so you shouldn't have two BBs, you shouldn't have AC. If you do that, and I say that because sometimes the numbers are very close and you may just pick the wrong ones, but this will be a double check on having done that. But let's say you've got it all correct. Now you notice over in the little purple box, I wanted to point out that when you see A plus plus, white, short, wavy, the genotype associated with that, in reality, that has the genetic component of AA plus B plus C. We simply left out that bottom homolog, that little a, little b, little c homolog, because it's the same all the way down. It's that early thing we showed you where we could simply drop them out. It's there, you have to know it's there, but we don't need it to work this problem. Now, what do we want to do? We want to determine the parentals and the double crossover. Well, that's very easy. By definition, the largest pair is my no crossover, or my parentals. We'll call them NCO. My double crossover, DCO, is the smallest pair. Again, it could be 0, could be none. These are, sometimes you don't actually observe any of these. Here we managed to see 12 of them, 6 plus 6, that apparently are part of the double crossover. Now, once you have that, you immediately use it to help you determine the order of these genes. That's what you want to do, is determine the order. By doing that, you're simply going to use the no crossover with the double crossover. Now, let's look at the idea of order for just a moment. If we have 3 loci, they could occur in 3 different orders. You could have B in the middle between A and C, so the order would be A, B, C. A itself could be, the locus could be in the middle, the curly locus or whatever. Could be it could be in the middle. It could be BAC, or we could have the C locus in the middle, which is ACB. Either one of those three. Now, we don't care which end at this point we read from. We don't care if we read it as CBA or ABC. It will matter in lab where you're trying to map, but for working general problems, we don't care. As long as we get the correct locus in the middle. Now, how do we get that in the middle? How do we determine if that's, we've written them down? We've written them down as ABC, but was that the right order or not? Well, that's a simple comparison of your no crossover to your double. Always start with your no crossover. Look at the difference between the two. Okay, I've pointed out the one thing that appears to be different here is that the A allele has moved from being A++ to now be associated with B and C. That one allele has moved. We know by definition that it was involved in the double crossover. Now listen to that carefully because this is where people get confused. The one locus that's involved in the double crossover will be the one that moves when you compare the no crossover to the double. Wherever it is, it could be on the ends or it could be in the middle. In this case, it's on the end. What that means is we've written this down in the wrong order. The true order is not A, B, C, but in fact the true order is BAC. The A is actually in between the B and C locus, and we know that by this direct comparison. So now what you have the correct order, and you should start to write these down in the correct order, is that A actually belongs in the middle. If we were to rewrite both of these genotype combinations, they should be plus A plus B plus C for the no crossover, plus plus plus, which doesn't change, but plus AC for the double crossover. Now that we've decided that they were not in the correct order, we need to rewrite everything in the correct order. We need to make sure that these will work. So here they are in the original order. We now know that A is in the middle, so I literally just pick up the entire A column and move it over. So in the first one will be A will be moved over so that we have plus A plus, and then we'll have B plus C. You can see how we will work our way down. There's the A being moved over. There's the next one being moved over. And then finally, the last two are moved over so that we have it all. It only takes a minute to do this little step. If you do not do it, it is very difficult to look at these and determine the right order. One of the biggest problems that we see in people having difficulty at this stage getting the final answer correct is that they fail to rewrite them at least in the correct order. All right, we now have them in the correct order. We can forget the old ones. We don't care about those. We now know what our no crossover is, our double. We have two single crossovers. You don't care what you number them. As long as it's 1 and 2, you keep them separate. Okay? Now we just simply need to do a little bit of very simple math. We want to calculate the map units for both the double and both of the single crossovers. So there's the double crossover. We had a total of 12 progeny out of a total of 200. So that's 6% or 6 map units or 0.06 as a fraction. We want to have then 20 plus 20 is the second form here, which is our single crossover number 1 divided by 200 is 20 map units. And then finally 28 plus 32 is 60. So that is divided by 200 is 30 map units. Now that we've gotten those, we want to use them to build our final model for the order of these particular genes on this chromosome. So what do we start with? We always start with the no crossovers. That's what we want to compare. Okay, so just grab those, pick any one of the singles, it doesn't matter, and just grab one of the singles and compare it back to the no crossover. So I'm going to take single crossover number 1 right here, and I'm going to compare it. I'm just going to move my no crossover down for you. You don't actually have to do this, but if you're uncomfortable with doing these, rewrite them right next to that single crossover and just take a look. What are we looking for? We're looking for one end or the other to move. Either the B or the C has to change position because we have a single crossover on one side of the A or the other side. Now we've got it in the correct order. You can see now why it's important to write it in the correct order. Okay, so what's different? Well, if we start out with plus A plus and B plus C, that was where we start, there's no crossover, and we end up with plus AC, we can see that the C locus, the one on the right side, has moved. That means the crossover must have been between A and C. Therefore, if we look at this now, we can see that the C has changed in form, which means that we want to put the data, the distance that relates to that single crossover between A and C. We simply write it down between A and C. There are 20 map units, or 0.20 at this point. This is easy between there. We also, as you notice, include a 0.06 because that represents the double crossover, because we know that double crossovers also occurred between A and C. Since we're trying to estimate the distance based on the number of crossovers, we might as well include those data to make it more accurate. So now we know that the true distance between A and C is 26 map units. Now the other side, you can repeat the same thing. You can draw down the no crossover to the other single, make a comparison. That's the safest way to do it. If you're in a hurry, you can just simply write down what you have, but I always suggest that you, just as a backup, compare the second single crossover back to the no crossover and make sure that the B, in this case, side is the one that moved, and in fact it did. So in this case, you would write down the other single crossover value, again, plus a double crossover, because that was also involved on this side. Write those down. We get a map units of 36. We now have our order, and we have the relative map units between these two. And as we said early on, this is one method that you can greater than 50 map units. You can see that now we know that A is between B and C, that the distance between A and B is 36 map units, and the distance between B and C is 62 map units. Had we done a simple two-way cross, or a normal mapping cross with just two loci, we wouldn't be able to tell that B and C, they would look like they independently sorted because they're greater than 50 map units. By using this method, we can literally walk down the entire chromosome and placing individual loci where they belong with great precision, which is what you're going to do in this week's lab exercise. And the last thing I want to mention is that you really cannot learn three-point mapping from any type of lecture, from simply watching it be done. You have to work problems. That is the one way. And for some of us, it's going to take a lot of problems. Some people will figure it out very quickly. As you don't worry about that, but do work problems. We'll reinforce this by having you do some in lab, but you need to try all the problems that you can find relative to three-point mapping. Practicing these problems is the only way that you're going to be able to do them when it comes to lab or when you come into lecture. So please set aside and plan on having some time that you can work these. If you can't work them, seek out help. They're